Hello, my name is Tristan Arroyo, and today I'm going to be talking about co-immunoprecipitation and immunoblotting. So, co-immunoprecipitation and immunoblotting are immunotechniques that target protein-to-protein interactions. Now, both of these techniques are used together, or they can be used together to exploit the strong and specific interactions between proteins and antibodies. Now, co-immunoprecipitation is a form of immunoprecipitation, which is used to isolate and detect a targeted or a protein of interest. The difference with co-immunoprecipitation is that it targets a protein complex, which means that it not only isolates your protein of interest, but proteins that are binded to it. Now, immunoblotting, also known as Western blot, is used to verify and quantify your isolated proteins or the complex. Now, co-immunoprecipitation is, I guess, not so difficult in its procedure or protocol. So the targeted complex is captured using specific antibodies for your target of interest. Then the complex is precipitated using a resin that contains IgG binding proteins. And finally, you wash off excess proteins that are not needed. And then your protein is ready to be analyzed. And that means you analyze it using immunoblotting. Now, the first step in immunoblotting is gel electrophoresis, and the point of this is to separate your proteins. So this separates your complex, pretty much. Then the separated molecules or proteins are transferred or blotted, per se, to a second matrix. And this matrix is usually a membrane composed of nitrocellulose or polyvinylidene difluoride, also known as PVDF. So this membrane is then blocked. So this means that you prevent non-specific binding of antibodies to other proteins. So you just want these antibodies to focus or bind, per se, or interact with the proteins that were blotted or transferred to this matrix membrane. So the transferred proteins then are probed with two antibodies. You have a primary antibody, which interacts with your protein of interest, and a second antibody, which contains an enzyme that binds. But this secondary antibody is like a complex in itself. It contains the enzyme, but the antibody binds to the host species of the primary antibody. Now, the importance of the enzyme is that it binds to a substrate, specifically a chromogenic substrate that is added after, that causes a color change or reaction. And this color change correlates with the protein. So when you see this color change, I guess you can tell that the protein that you're looking for was actually present in this membrane where it's been transferred. So now a real-world example of co-immunoprecipitation and immunoblotting. So this was taken from an Alzheimer's disease treatment paper. And here they asked the question for this specific experiment, the specific experiment using this figure of by which mechanism does sodium selenate reduce tau phosphorylation? Now, I guess a bit of background on Alzheimer's disease. So there's two forms of pathogenesis that have been, I guess, known as now. One of them is amyloid beta plaque formation, and the second is tau phosphorylation. Now, tau becomes hyperphosphorylated and creates these neurofibrillary tangles in the brain, which is associated with neurodegeneration, which is associated with Alzheimer's disease. Now, here, the reason they are using sodium selenate to reduce tau phosphorylation is because sodium selenate contains selenium, which is an element found in the brain, and low levels of selenium have been linked to, cognitive impairment and Alzheimer's disease. So their thought process here was, by increasing the amount of selenium, can they reduce tau phosphorylation? Now, here the design here was that they took tau from the cell culture of SHSY5Y neuroblastoma cells, and they took it from this specific cell culture because it contains a form of human tau or phosphorylated human tau, and they can't specifically, extract this from human brain since you can't take that many neurons. So the techniques here are co-immunoprecipitation and immunoblotting. So the purpose of the test was to test for the presence of PP2AC, which is a catalyst that they believe, or I guess found, to bind to tau when it's phosphorylated and sort of dephosphorylates it. But what they found is that it only binds and forms this mechanism to tau when with the presence of selenium. So that's why they're adding selenium here to tau to see if this is correct, to test their, I guess, hypothesis that PP2AC is selenate, I guess, dependent. So that was their outcome, to expect to see PP2AC bind to tau when it is treated with selenate. And that's what you see here in figure A. So here you have, the tau with the presence of selenate. So here in the first line is a negative sign, which means you don't have any selenate. And you, as you can see in the first band, so this is the results of the immunoblotting. In the first band, you see HT7, and you see 3 very strong bands. And this HT7 is an antibody for tau, for human tau. So this just shows, or means, it's like a control per se to know that you have tau in all 3 of these forms, or all 3 of these different experiments, or different trials per se. So it's just showing you that tau is present in each one, without selenate, with selenate, and with an even higher concentration of selenate. Now, the PP2AC here, that those bands are the presence, are to show that PP2AC is present when selenate is added or treated to, when tau is treated with the selenate. So as you can see, they increase the concentration of selenate, and you can see the increase in the abundance of PP2AC with an increase in selenate. So I guess some limitations and strengths to this experiment. So some strengths were that it kind of gets closer to a treatment or some form to battle the pathogenesis associated with Alzheimer's disease. But I guess a strong limitation to that is that it's not sure per se to completely stop or eradicate Alzheimer's disease. It just kind of slows it down or fights the phosphorylation. And it didn't target anything with the beta amyloid plaque, which is also a very big deal associated with Alzheimer's disease. Now, some questions I had. So one question was, it's about the more on the co-immunoprecipitation part. And is it possible to detect or target proteins without a specific antibody? Since we know that you use specific antibodies to these proteins when you want to target it. So I want to know if it's possible to isolate or target a specific protein without having an antibody. And then here are my references and the end.